Hello, I'm Lauriane Mietton. I'm currently in my third year of PhD in microbiology in the SPO lab at INRAE, France. With this poster, I will present you how, with a participatory research involving bakers, millers and researchers, we demonstrated that bakery environment shapes sourdough microorganisms community and bread aroma profile over milling technique. Sourdough bread is made out of flour, water and sourdough. Sourdough is a fermented mix of flour and water. In this medium, microorganisms, yeast and bacteria develop to form a stable microbial community. In order to keep this community active and fermentative, flour and water are regularly added. This operation is referred as doing backslopings. To obtain the flour used to make both sourdough and bread, wheat is firstly milled, and different milling techniques exist to produce flours. The most commonly used by farmers, bakers and bakers are stones mills and rollers mills. So we decided, together with the group, to compare these two techniques. Our common scientific question was how milling impacts sourdough community dynamic and bread quality. To answer this question, we built together an experimental and participatory design involving 6 millers, 4 bakers, 2 master students, and a whole research team with engineers, technicians, and researchers. We picked 4 different wheat mixes that we sent to 6 millers, 3 Wistons mills, and 3 roller mills. We obtained 24 flours. We then randomly distributed 6 of them to each of the 4 bakers, making sure that each have half roller mill flours and half stones mill flours. They initiated 7 sourdough in their bakeries with these experimental flours together with their own usual flour, and they backsloped them for all around 1 month. At the end, breads were made with the sourdoughs and their corresponding flours. So at first, we characterized flours parameters. Then we analyzed sourdoughs all along the month of backsloping in terms of microbial density and specific composition. At the end, we analyzed bread proteins and sugar content and bread aroma compound ratios. I will present you 2 of the main results. First, the dynamic of sourdough community establishment according to microbial analysis. In this figure, you have a similar representation for yeast on the superior part and lab on the inferior part. The colors represent different microorganism species. In rows are put the 7 sourdough of each baker, AM, MB, PDC and LB. As a remainder, 3 sourdoughs were made with stone mill flours, 3 with roller mill flours and 1 from the usual baker flour. In columns, you have the number of the backsloping corresponding to the sampling time. T1 is 24 hours after the initiation, just before the first backsloping, T5 just before the 5th, and T32 just before the 32nd. For yeast, as for lab, a great diversity of species observed at the beginning, diversity which plummets at the end with only 1 main yeast species remaining and 1 to 3 main species remaining. As a result, observed for all sourdough. Establishment of main live species is slower than main yeast species that appears as soon as the 5th backsloping. Finally, it is well observed that milling did not impact the dynamic and composition of microbial main species establishment, contrary to baker that shaped this composition. Another result is the bread aroma profile. This analysis revealed that milling of the flours has no impact on it, contrary to sourdough community specific to each baker. This heat map links bread made by each of the 4 bakers and aroma compounds grouped in 9 chemical classes. The color intensity of each square represents the intensity of the quantity of the bread aroma compounds belonging to the classes. In conclusion, 3 results of this participatory project are first, that milling has no impact on sourdough microbial dynamics and bread organoleptic quality. Second, for all sourdough, 1 to 3 main species of both yeast and lab finally dominate the microbial community. Third, bakers have their own specific sourdough community that drives bread organoleptic quality. If you have any question about this work, I will be pleased to answer them. Thank you very much.